This video is a test review video for meiosis and mitosis. There are going to be more meiosis questions, but there will be some comparison. Remember that if you don't understand the difference between meiosis and mitosis, you really don't understand either process. This screen shows the important vocabulary words from this unit. Variation has been a very important concept in this unit. And why do we care so much about variation? Imagine a disease that kills organisms with a trait for green color. Which of these populations would be more vulnerable, the one on the left or the one on the right? The answer is the one on the left. In that case, all of the green individuals die out, and in the population on the right, you have some individuals that survive to reproduce. So with increased genetic diversity, a population can better survive environmental pressures. Which gives more genetic diversity, sexual reproduction or asexual reproduction? The answer is sexual reproduction. The three additional sources of diversity in sexually reproducing species include random assortment of homologous chromosomes, crossing over, and random fertilization. Remember that asexually reproducing species do have one source of variation, and that is mutation. This diagram shows you a diagram of the regular cell cycle for mitotically dividing cells. In the first cell, we would have single-stranded chromosomes that are, the cell will be diploid, so it would be 2N. And then DNA replication would take place during the S phase of the cell cycle, and that would mean that we have double-stranded chromosomes, and we would still have 2N cells because the cells are still diploid. And then when mitotic cell division occurs, we would have single-stranded chromosomes, but we would still have the diploid number of chromosomes in the cells. Remember that in mitosis, there is no change in chromosome number. You only have a change in the amount of DNA between the different phases of the cell cycle. So the amount of DNA doubles after the S phase, but divides in half after mitosis. So when you see this diagram, every time you see SS, that means single-stranded. Every time you see DS, that means double-stranded. This diagram shows meiosis. Your first parent cell, your primary spermatocyte or your primary oocyte, would be diploid. And the chromosomes would be single-stranded if it's before DNA replication. DNA replication has to happen before meiosis and also mitosis. So after DNA replication, you would have a double-stranded chromosomes in your cell, and your cell would still be diploid. Now, after meiosis 1, the cell goes from diploid to haploid. Remember that homologous chromosomes separate during the first division in meiosis. But we still have double-stranded chromosomes. Meiosis 2 separates sister chromatids, so we go from the double-stranded N number of chromosomes to single-stranded. So our gametes would be haploid and single-stranded. But remember, the transition from diploid to haploid happens in the first division, not the second division. The second division is to separate the double-stranded chromosomes into single-stranded chromosomes. So this animation shows you synapses and disjunction. Synapsis is when the homologous pairs find their mates. Disjunction is when those homologous pairs separate. Disjunction happens in meiosis 1. Now these two diagrams show you two sources of genetic variation in meiosis. The process on the left, what is it? That's showing you random assortment of homologous chromosomes. Remember that your chromosomes can line up differently. They always line up with their homologous pairs, but which chromosome ends up in which gamete varies each time. The process on the right, what process is that? That's crossing over. It's sometimes also referred to as genetic recombination because you get new combinations of traits on chromosomes. What do these processes have in common? They both add genetic variation to sexually reproducing species. In other words, new combinations of genes. So One could consider random assortment of homologous chromosomes to be genetic recombination, but generally when you see genetic recombination used, the term is usually referring to crossing over. What is the only source of genetic variation in asexually reproducing species? Mutation. Can this also create variation in sexually reproducing species? Yes. There's more genetic variation in sexually reproducing species. How is this an advantage? Well, it makes the species more likely to survive if their environment changes. At least some of the individuals would have traits that would allow them to survive better in the new environment. Those traits, if they help them survive better in the new environment, would be considered adaptations. Meiosis is the production of gametes, sperm or egg. What change in chromosome number happens during meiosis? A diploid cell produces 4 haploid cells, so you go from 2N to N. The chromosome number divides in half. In order to have sexual reproduction, fertilization has to occur. It's not enough to produce gametes. Fertilization is when the nucleus of a sperm fuses with the nucleus of the egg. The scientific term for egg is ovum. Plural would be ova. What change in chromosome number occurs during fertilization? 2 haploid gametes fuse to make a diploid 2N cell. That fertilized egg is called a zygote. After fertilization, a zygote begins to divide rapidly. What type of division is this? Mitotic cell division. Is there any change in chromosome number in mitosis? No, the zygote is diploid, and so are all of the cells that result from mitotic division. If there are 112 chromosomes in the sperm of a moth, how many chromosomes would there be in the cells in its wings? There would be 224, because if the sperm has 112 chromosomes, the egg would have 112 chromosomes, and when fertilization occurs, you would add those together to get 224. The cells in its wings would be somatic cells or body cells. So let's compare spermatogenesis and oogenesis. Spermatogenesis is production of the sperm, and oogenesis is production of the egg. The chromosome number is exactly the same within a species in sperm versus the egg. In humans, sperm have 23 chromosomes and eggs have 23 chromosomes. In terms of the number of gametes produced, sperm production produces 4, and in egg production you have 1 egg and 3 smaller cells called polar bodies. The job of the polar bodies is to just break down after removing excess genetic material. Sperm are small and equal in size, whereas eggs have to be large. The reason eggs have to be large is because when they divide, there's not very much time for the cell to grow in volume between cell divisions. So if you start off with a lot of cytoplasm and a lot of organelles, it's easier for the cell to divide rapidly. The sperm have one job, and that is to swim to the egg and fertilize it. So they must be motile, and they use a flagellum to swim. Eggs are non-motile. They do not move on their own, but there are cilia in the fallopian tubes that create a current that the egg floats down and that the sperm must swim against. The function of polar bodies, again, is to remove all of the excess DNA while leaving behind as many organelles and as much cytoplasm as possible. Meiosis and fertilization together give us sexual reproduction. Let's review our prefixes. Mono means 1, di means 2, tri means 3, and tetra means 4. The term haploid refers to having 1 of each type of chromosome. So you do not have homologous pairs in a haploid cell. If you had a triploid cell, you would have 3 sets of chromosomes. A diploid cell would have 2 sets of chromosomes, and a polyploid cell would have many sets of chromosomes. There are plants that survive completely fine with polyploid nuclei. A chromosome is a thread-like structure within the nucleus containing genetic information that's passed down from one generation to the next. A replicated chromosome is double-stranded and has two identical sister chromatids that result from DNA replication. Homologous chromosomes are corresponding pairs, one from the mother and one from the father, that code for the same traits and have the same structure, including size or length, shape, and shape is determined by the location of the centromeres and the banding pattern. Now, it's important to note that homologous chromosomes, when we say they have the same traits, it doesn't mean they're identical. So, for example, if you have two homologous chromosomes, one might have a gene for blue eyes and the other might have a gene for brown eyes. So they're coding for the same trait in terms of the idea that they code for eye color, but they may not be identical in terms of their alleles. So we call the different variations of a gene alleles. So you have brown-eyed alleles and blue-eyed alleles in this particular scenario. A gamete is a sex cell. It is a general term that includes sperm and eggs, and it results from meiosis. Gametes have half the number of chromosomes of any body cell from that species, and we use the symbol N to refer to haploid cells. Somatic cells are body cells, and they include any cell in an organism that is not sperm or egg. Somatic cells are always diploid, and we use 2N to represent the idea of diploid cells. So 2 times the N number. So in humans, the haploid number is 23, and the diploid number is 2 times 23, or 46. So for this slide, we're going to be looking at cells and deciding whether they are haploid or diploid. There is one thing and one thing only to look for. You're looking for the presence or absence of homologous chromosomes. So in cell number A, there are 4 chromosomes, but none of the chromosomes are the same size as each other. So we would say that that cell is haploid. There are no homologous pairs shown, and each chromosome has its own unique shape and size. Look at cell number B, not cell number B, cell letter B. And in cell B, you see 2 chromosomes of each size. You have 2 small, 2 medium, and 2 large chromosomes. Therefore, cell B is diploid. There are homologous pairs shown. Notice that they are not double-stranded. Some students confuse the term diploid and double-stranded. Just because something is single-stranded doesn't mean it's haploid. Cell C has 3 chromosomes. If you have an odd number of chromosomes, the cell can't be diploid. So this one's a little bit more obvious, but still you have a small, a medium, and a large chromosome, and 1 of each type of chromosome. So that cell would be haploid. There are no homologous pairs shown. There's also an odd number of chromosomes. Cell D has 1 of each shape, so that one is haploid. No homologous pairs are present, whereas E, we have 2 circles, 2 squares, 2 triangles, and 2 squiggles. That cell would be diploid. There are homologous pairs. And in cell F, we have 2 large and 2 small. These 2 are homologous, and those 2 are homologous. So that cell would be diploid. If a cow had 60 chromosomes in its somatic cell, how many chromosomes would be in the gametes? There would be 30. And during fertilization, they would fuse, and the zygote would have 60. If a diploid cell has 16 chromosomes, how many chromosomes will be in a haploid cell of the same organism? There would be 8. For these following cell types, we need to identify whether they would be haploid or diploid. A sperm would always be haploid. Muscle cells would be diploid because they are body cells. 2N represents diploid cells, N represents haploid cells, and zygotes would be diploid. It's important to note that chromosomes are not considered haploid or diploid. Cells are considered to be haploid or diploid depending upon whether they have pairs of homologous chromosomes. Any mitotically dividing cell would be diploid. Homologous chromosomes separate during meiosis 1, while sister chromatids separate during meiosis 2. So if we look at this cell division and we look at our chromosome pairs, how many possible combinations of chromosomes would there be in the gametes due to random assortment of homologous chromosomes? The way to figure this out is 2 to the n number. Our diploid number is 6. Our haploid number is 3. So 2 to the third power would be 8. So now let's compare mitosis and meiosis. In mitosis, the cells divide once. Meiosis has 2 rounds of division. At the end of mitosis, you have 2 daughter cells. And at the end of meiosis, you have 4 daughter cells. The parent cells in both cases are diploid. The daughter cells at the end of mitosis are diploid. The daughter cells at the end of meiosis are haploid. And the reason for that is that in meiosis, homologous pairs separate. They do not do that in mitosis. At the end of mitosis, you have genetically identical cells. And at the end of meiosis, you have genetically different cells due to random assortment of homologous chromosomes and also possibly crossing over. At the end of mitosis, you have somatic cells or body cells, and at the end of meiosis, you have gametes or sex cells, sperm or eggs.